So thank you for having me here. I'm Russell Schwartz from Carnegie Mellon University. So I can say I am formally trained as a computer scientist, but I've been doing nothing but computational biology since I was an undergraduate, so I consider computational biology my area. What I want to talk about is some problems in tumor phylogenetics, and really focusing very specifically on methodological questions. I know some others at this meeting will be talking much more about what these methods tell us about the biology. I want to focus on some questions about the methodology. And this probably doesn't need much explanation for anyone at this talk, and thankfully Ben gave a lot of the background. I can now skip over to some degree. But what we're working on, what many people in the field are working on, is really guided by this notion of cancer as a disease of evolution. And from our point of view, the key thing that you need to know about cancer as a disease of evolution is that for the most part, tumorigenesis begins from a single mutated cell that usually acquires some kind of hypermutability defect that leads to a rapid accumulation of mutations that produces rapid diversification in the progeny of that cell, and diversification gives an opportunity for selection. So for selection for particular mutations that give the tumor cells an advantage in growth rate or escaping normal controls on cell growth, and that combination of diversification and selection is evolution. That's what is guiding in our understanding of how cancer works, how cancer progresses. All of that has been known for many decades at this point, but what really changed with computational biology is taking that from a kind of guiding principle for how we think about tumor development to a source of practical methods that have really become crucial for how we interpret tumor genomic data. Essentially, without this kind of understanding, we would not know what to do with tumor sequencing data. And to a large degree, that's come through the problem of, or the strategy of tumor phylogenetics, the idea of bringing phylogeny algorithms to understanding tumor genomic data. This idea originally started with what were called oncogenetic tree models, which were essentially a way of looking at individual tumors, modeling these as if they were species within an evolutionary tree, and by building a tree of those different tumors, get an idea of how those tumors partition into distinct subtypes and what some of the major decision points of that were. So what were the particular patterns of mutations that lead tumors down different paths of these oncogenetic trees, which can have important implications for diagnosis, prognosis, treatment, and so on. My own lab and others got into the field with a different view of tumor phylogenetics as a way of building phylogenies of single tumors, in our case based on cell-to-cell variations within these individual tumors, of variations between single cells in the tumors, and by building a phylogeny of those single cells, come up with a model of how one tumor is developing. And one of the key things you learn when you start looking at tumor phylogenies is that one phylogeny tells you absolutely nothing because it's mostly random noise, but once you start doing this at scale, you can figure out what is actually meaningful and robust about these individual phylogenies and start to learn something about the biology of these tumors. And I mostly want to talk about methods that are focused on phylogenies of single tumors, although many of them actually branch back to these oncogenetic tree models, so essentially bringing in pieces of understanding variation tumor to tumor, so intertumor heterogeneity, and variation within the tumor, intratumor heterogeneity. And this probably doesn't need to be said to this audience, but this has gone from a fairly specialized topic that only a few people were working on maybe a decade ago to really part of the mainstream toolkit of cancer research. So these are a few papers, so I guess this is one of Roland's papers, but these are papers that were primarily experimental studies, but where a phylogeny now is a key part of how you present and interpret that data. And this is something I think no one in cancer research today would be surprised to see in a Nature paper, so a phylogeny of a tumor showing up. 10 years ago, that would have been a pretty surprising thing and needs some justification. I think this has been a great area for computational biology because of that. But what I want to do is talk about some methodological directions, as I mentioned. And to understand that, it's important to know something about the different kinds of tumor phylogenies that are available. There are a lot of different ways you can break down what can be done with tumor phylogenetics, and one way of thinking about it has to do with different sources of data available for building phylogenies. My own lab's work got into this using what I refer to as pre-genomic technologies. So tumor phylogenetics really predates the genomic era. And pre-genomic methods, in our case, are primarily fluorescence in situ hybridization, or FISH, which gives you information about a lot of single cells, but very little information about each cell. And that turns out to be a really useful thing for studying tumor phylogenetics because it lets you work at scales where you can see a lot of variation in single tumors and do this for enough tumors to start cutting through that noise, the inter-tumor heterogeneity. So this was historically for us an important technique. We're still working on this and still learning things that I think you can't learn from any of the other techniques here. But for the most part, this is a fairly specialized method. So it's not the main thing I want to talk about today. For the most part, when people are working in tumor phylogenetics, like Ben was showing, they're working with bulk sequencing data. This might be a single sample per patient, it might be multi-region sequencing, but most of what's being done in tumor phylogenetics so far is coming from looking at these bulk samples. And Ben saved me a lot of time explaining some of the trade-offs in working with this kind of data, because bulk sequencing has huge advantages over the pre-genomic technologies in allowing you to see, in principle, the entire genome. It's been done at much larger scales. Of course, there are cohorts now of thousands of patients who have had bulk sequencing of their tumors. That's a really useful thing if you want to figure out what is robust and predictable from these tumors. But it also has some significant disadvantages to a large degree because of these deconvolution problems Ben was mentioning. So if you are working with bulk sequencing, you are mixing many different genetically distinct cells in each sample. You have to separate those. That is often a hard computational problem, and it can only be done with limited resolution. So the trees you get from bulk sequencing are much simpler than the trees you get from true single cell data. We know there's a lot more going on there than we're seeing in a deconvolution. Hopefully you're getting the major features, but it's always important to remember that you're seeing a coarse-grained view of the clonal heterogeneity within these tumors. Because of that, we and many people in the field are quite excited by single-cell sequencing, and that in just the past few years has really emerged as its own area of tumor phylogenetics. So I think some within this room are working on that. My lab is working on phylogenetics of single-cell sequencing as well, but for the most part, that's not what I want to talk about today either. I mostly am going to be focusing on problems related to bulk sequencing. I do always like to say a bit about the FISH data because we're one of the only groups who's working on that, and because I think there are important lessons that guide our thinking and are useful for other thinking in the field. But at a very high level, what FISH is simply a way of fluorescently labeling particular regions of a genome for the purposes of tumor phylogenetics. It's useful because it gives you data on lots of individual cells, although, as I mentioned, very little data per cell, a few copy number variation markers per cell. But because you can get these for a lot of cells, you can get very fine-grained views of clonal heterogeneity within these tumors on those markers. You can use those to build quite involved phylogenies. So these are not showing up very well, but so the details are not very important for this talk. And you can do this on enough scale that you can start to mine these for information about variation tumor to tumor, so to reconstruct patterns of variability, so differences in the evolutionary process itself as opposed to just differences in the end state of that process, tumor to tumor, start to pull out different mutator phenotypes, so different preferences for mutability tumor to tumor, which we find are really one of the most interesting and important sources of information if you're trying to figure out what these are telling you about tumor development, and show through a variety of different ways that you can take that information and use it to not just reconstruct the past of a tumor, but also make accurate predictions about the future or progression of that tumor. And what I want to leave you with before I move on to really the main topic of my talk is some of the key lessons of that are guiding my thinking, and I hope I'll be able to revisit at the end of this talk. So one of the key things we learned is that tumors are highly heterogeneous. This was, of course, known, but this became apparent very early in these FISH studies back when we first first got into this, that the thinking about tumor evolution before you could really look at it was assuming much simpler models than what is actually going on. So there were great thought experiment models, Fearon, Vogel, and so forth models of more or less linear progression of tumors, and what you see when you actually look at tumors is a lot more complicated, and it ends up being quite important. If you really want to see what's going on, you need to look at a lot of clones and a lot of tumors, and you will discover that most of what's going on there is noise, or at least random chance. Now, there's a great talk by Quaid at ISID a few days ago where he made the very important point that you can learn a lot from noise, and that is definitely true if you are trying to understand the mutation process. So I don't want to belittle noise here, but most of what is there is noise, and it's important to understand that. And you really need to understand what's going on in the fine details, because when you look at progression processes, sometimes progression proceeds from major clones, sometimes it proceeds from minor clones, sometimes it proceeds from clones so minor we don't see them, even profiling hundreds of single cells per tumor. So it's important to know that you really need to understand what's going on in the weeds if you want to understand how the tumor is behaving or what's driving the next steps. And one of the things you can pull out from this that's quite important is that diverse mutation processes are at work. They vary tumor to tumor, they vary from time to time within a single tumor, and a lot of the interesting information about tumors has to do with not the particular pattern of driver mutations a tumor has at an instant in time, but the mutation processes that are leading to these drivers. And that's a lot of what is guiding our thinking about what we want to look at in tumor evolution studies. But all of that is kind of a prelude to another line of our work, and really more of the work of most people in this field, which is working from the bulk genomic data like Ben was telling us about. So with bulk genomic data, as you've already heard, you were looking at bulk mixed samples, and essentially the problem is if you want to build a tumor phylogeny, you have to deconvolve those samples, separate them into cells that are mixed to different degrees in different samples, and then you can build phylogenies, or concurrent with deconvolution, you can build phylogenies. So that essentially is the problem we want to work on there. And my lab has been working on this almost as long as we've been working on the fish data sets, but I would say this is a much harder problem in some ways. It's a problem where I think there are a lot of things still unsolved, and so I want to say a little bit about some of the methodological questions and some of these still unsolved problems we're trying to solve there. A lot of our work was based on the notion that you can understand mixtures in terms of models from geometry. As near as I can tell, this idea first came from these people who were working in geology, so they were not working on anything related to cancer or genomics at all, but essentially their key observation was that you can understand the mixed membership problems, the problem of blind source separation or unmixing. There are about a dozen different names, this is known by in different fields, as a problem in geometry. And the big idea behind this is simply, if you have a set of mixed samples, like two fundamental cell types, you plot them in some genomic space, so gene copy number space or expression or whatever, then mixtures of those lie on line segments between those points, and if you can find the line segments, you can deconvolve your mixtures. If you have 3 components, you get triangles. If you have k components, you get k-1 dimensional simplices. So essentially, finding geometric structures in high-dimensional space is equivalent to the problem of deconvolution. Now, it ends up, as with most things in genomics, not really being that simple. You can get reasonably far by just solving these geometry problems, but it's a very noisy representation, in part because genomic data is always very messy. In practice, one of the first steps we ended up taking was realizing you can't just solve the geometry problem. You need to solve what we ended up calling a soft rather than hard unmixing problem, which basically means try to figure out the kinds of noise you get in genomic data, build some tolerance for that into the model. That ends up leading to some significant improvements in accuracy, and really led to, for us, some of the first methods that were really useful for deconvolving some of these data and building phylogenies from deconvolved data. This was an early version of this from our work some time back, from an even earlier data sets. This was some early work from Nick Navin's group, This was looking at array-based copy number phylogeny or array-based copy number data. And essentially the key point of this was that you could take some data and through a kind of combination of the oncogenetic tree modeling and the single cell modeling, come up with a very coarse-grained representation of clonal heterogeneity. Essentially what we were trying to do, because data was fairly scarce in those days, was take similarities among different tumors as well as heterogeneity within tumors and try to build a kind of cellular tree like Ben was talking about, where different steps in that tree would correspond to approximate clones that would be visited by multiple tumors over similar patterns of progression. So essentially we would propose this as a kind of oncogenetic tree where different tumors are proceeding down different subtrees of this tree. And of course that's very coarse-grained, very approximate because they're all doing different things, but you can get rough models of, in this case, copy number profiles of cell states that are approximately visited as you progress through these trees, build a coarse-grained phylogenetic model of these, and get something that at least gives you a handle on the variation. Yes, that's an important point. Although I'm calling this tumor phylogenetics, it's not properly phylogenetics. We're using phylogeny algorithms and tools, but we're using them for a problem that's not, strictly speaking, a phylogeny problem. So thank you for pointing that out. All right, but basically this is what we were able to get to with an early version of these kinds of methods. But as I'm trying to emphasize, it's a very approximate version of this problem, and that's really a starting point. And what I want to talk about for whatever time I have left is mostly how you get from that starting point and lots of different directions you can go to get closer and closer to the reality. And I want to emphasize here that we know the reality is much more complicated than we're getting from any bulk deconvolution method right now. So this is not about getting to the ground truth, it's about moving us a little closer in various directions to that ground truth. And there are a lot of ways you can do that. So if you know you are getting an answer from your data that is an imperfect answer, it's not quite the right answer, then there are various ways you can try to improve on that. You can try to get better data, you can try to get better models, you can make your models better mathematically, you can make them better through better biological knowledge. And one of the things I hope you get as a takeaway from this talk is that every piece of that is important if we're trying to improve tumor phylogenetics. One of the ways we went about trying to improve this is by recognizing that the mixtures we get from tumors are not really quite what we would expect from those simplicial models, the simple geometry models. And you can see this when you look at a more modern data set right here. So this is a couple of subsets of the TCGA data, ovarian, lung cancer. These are both copy number variation data sets. And you can probably see in PCA space, there is some geometry to these. There's some structure, but they decidedly do not look like simplices. We're not seeing triangles or tetrahedra or whatever. And if you use your imagination a bit, you can kind of see that there are one-dimensional projections here, maybe two-dimensional spaces. We can maybe say that this kind of looks like a couple of one-dimensional pieces glued onto a two-dimensional piece. This looks like a couple of distinct two-dimensional wings projecting out from a single point here. They look like not simplices, but what are referred to as simplicial complexes, collections of simplices in lower dimensions that are joined together. And this actually makes a lot of sense if you think about the oncogenetic tree models. If we really believe that all of our tumors are approximately following some different directions that a tumor can evolve down, but different tumors are sampling different subparts of this tree, then we would expect that what you would see would be a simplicial complex. Tumors that are sampling from this subtree would form a low-dimensional manifold based on the cell types in this subtree. Tumors sampling from this one would form a different low-dimensional manifold, and those manifolds would be glued together by the cell types they share in common. So this is a mathematical model, as with everything in biology, the model is not quite the real thing, but it's somewhat closer than simplices to the model we actually want. And essentially what we can do is try to refine our problem to say that this now is what we're looking for. We're really looking for simplicial complexes, not simplices. We can pose an improved model. So this is a variant on the kinds of geometry models we had before, where we're balancing essentially a cost of the deconvolution, a cost of the vertices. We end up charging the vertices based on an L1 minimum spanning tree, which is a very crude approximation to a minimum evolution phylogeny, and then some additional terms to bias it towards not overfitting the data to regularize and so forth, giving us a statement of what it is we're trying to find when we look for simplicial complexes. Now, sometimes being able to specify your problem is about as good as being able to solve it. You just throw your computer science at it and you've solved your problem. This is not one of those cases. We can specify what we're looking for, but it ends up being a very hard thing to solve for. So this is actually a special case of a class of machine learning called manifold learning, which is basically trying to discover structures in high-dimensional spaces. But it turns out to be a really ugly, bad example of manifold learning. In part, it's a bad example because we have very sparse data compared to what most people assume there. It's very noisy data. There's a very big gap between what's called the ambient dimension and the intrinsic dimension. So intrinsic is the dimension of the thing you're trying to find. Ambient is the actual space of the data you have, which, if it's genomic data, is anywhere from tens of thousands if you're looking at the gene level to billions if you're looking at the nucleotide level. So, and also in manifold learning, you usually would be assuming that you're looking for a smoothly varying shape in your space. In our case, we very decidedly have non-smooth shapes. We're specifically looking for shapes that have these discontinuities. And what all that contributes to is that this is a hard problem to solve, and we end up having to solve it heuristically and probably not to optimality. Basically, we end up coming up with a pipeline where you reduce your dimension, you use specialized kind of clustering to discover the subspaces, unmix the subspaces, try to resolve this into a simplicial complex. It's a crude method, but it gets the job done and allows us to do some discovery of the structure in these spaces. This is TCGA copy number variation data, and you can kind of see there's some geometry in the raw point cloud here. These gray lines are showing the computational reconstruction. It's very coarse-grained. It's probably not doing as good a job as you or I might be able to do just by eye, but it's getting at something in this data set here. And really what it's saying is an approximately very simple model of different subtypes of progression mixed with normal cells. We have impure progression along a few lineages. We know what's really going on is much more complicated. That's a starting point. We can show through a variety of tests that's concordant with standard immunohistochemical subtyping of these tumors, various other ways of deconvolving them, concordant with deconvolution from other data, all of which suggests that it's imperfect, but to some degree it's getting at a ground truth, and we can show that there is at least enrichment for various biological terms at the vertices of these that would be suggestive of it pulling out meaningfully different subsets of our cancer data. So that is telling us one thing about how we can improve on our methods. We can understand the structure of our data, and in this case particular, how mixed data behaves, and the fact that the kinds of mixed data you get from cancer are not the uniform mixtures you would predict from earlier models. They have some substructure you can exploit. Another way we can try to improve on this, as I mentioned, is improving your data. And it's nice when we work with these big consortium efforts like the TCGA that they have a lot of different data. And data can also help you if it is complementary to the other kinds of data you're working with. This is what we get applying more or less the same method to RNA sequencing data. And it also does a deconvolution. It's also not a very involved one. It actually looks like a really bad deconvolution here. It's not quite as bad as it looks because we're seeing a two-dimensional projection of a higher dimensional object, but it's not a great deconvolution. But it's a useful one because it ends up deconvolving to some degree orthogonally to what we see from the CNV data. We get, again, good concordance with immunohistochemical subtyping of these data sets, but when we look at term enrichment, one of the things that comes out very clearly is that one of the vertices we're pulling out is strongly enriched for immune terms. Essentially, we're probably pulling out a subtype here related to immune infiltration, which is invisible to the CNV data, but is very important for understanding the cellular heterogeneity in the tumor. And what that suggests is that maybe we can do better by bringing those two data types together. And to a degree, that is indeed true. If you bring in, with some normalization, the DNA and RNA into a single analysis, then we can get a more involved simplicial structure, a real simplicial complex coming out of this, where we can see a more involved subtyping. Actually, one that to some degree does not do as well by the statistical tests, probably because it's breaking down the data into finer categories, but ends up giving us much more specific, if less sensitive results when we look at term enrichment of the vertices. Essentially, it's much more specifically pulling out terms that are related to, in this case, breast tissue cancer or breast cancer, which suggests that bringing these data types together and relying on the complementary advantages of different sources of data gives us some benefit over looking at one data type alone. So that's telling us another way we can improve on this and still get very far from the ground truth, but kind of turn the knob a little bit, get an incremental advance. Another way we can do this is by trying to bring in other kinds of information we can extract from this data. And one of the things we've been very interested in is bringing in structural variations. Most of what my lab has worked on is copy number variations, and I do think copy number variations are a really important class of data, in part, as Ben was saying, because they're a significant confounding factor for all your other variations, but in part because copy number changes are a major, arguably the major way tumors actually evolve. They gain copies of oncogenes, they lose tumor suppressors. That's arguably the main way tumors actually adapt functionally. But it's important to remember that copy number variations are just a side effect of broader classes of structural variations. Insertions, deletions, inversions, duplications, and so forth. And so another way we can try to improve on this is to extend our models to bring in these other kinds of data. So in fairly recent work of ours, so just presented at ISMB about a week ago, we had asked, can we take these deconvolutional models? In this case, we're actually going down to single sample models rather than those multi-sample models. This technique works on multi-sample. We're looking at one tumor at a time, and try to ask, can we do more with information about classes of structural variations? In particular, leveraging the fact that the sequence data you would ideally use to get your copy number variation also contains breakpoint information you can use to classify structural variant events in the tumor development. Essentially, by seeing split reads, so reads that align to multiple points on a genome, you get information that's telling you about, at least to some degree, what structural variations are going on, and there are ways we can exploit this as well in our tumor deconvolution and phylogeny approaches. So we proposed to ask, let's see if we can improve on our deconvolution by bringing in that other source of data. We can start with actually a simpler model of deconvolution than we've been working with before, so just a very simple version of the non-negative matrix factorization approaches, and then throw in a phylogenetic cost like we've been working with previously. But this is a phylogeny where we're bringing in the structural variations. We're actually scoring the phylogeny by copy number variations, so again, using this kind of minimum evolution approximation, but we're adding in a perfect phylogeny on structural variation constraint, essentially saying that we believe breakpoints in the genome are rare enough that the same exact base level breakpoint should not be observed more than once, and then throw in the constraint that will only look for phylogenies that obey the infinite sites model with structural variants, although not with the copy number variants. Throw that in as a constraint, and then add in one more constraint that is basically asking, are the copy numbers of our breakpoints consistent with our model of copy number variation in the tumor, essentially penalized for deviations between read counts in our copy number models, and ask, does this help us improve our phylogenies? This also ends up being a problem where we can state what we're trying to solve, but solving it is another challenge. So we end up posing this as a fairly involved integer linear program. This is actually not the entire program. These are kind of the major pieces of it: a deconvolution cost, a phylogeny cost, a breakpoint cost. It ends up being too hard to solve to optimality, so we come up with a coordinate descent algorithm where basically you solve for your cell types, assuming everything else is known, then you solve for your deconvolution, assuming your cell types are known, and try to converge on an optimum this way. It's probably not doing so to optimality, and actually even this is not practical for some of the most highly structurally variant tumors, but it gives us something we can work with, where we can then start going into data sets and ask, do we learn anything by throwing in the structural variants? This is a view from simulated data, so just bringing in simulated phylogenies to ask if in principle this works. And the key point here is that it does give us a significant improvement in our ability to deconvolve. This red line here is what you get with deconvolution without these structural variant constraints. This is just a measure of error, which basically says it doesn't work very well. The green line is our method bringing in the structural variant constraints. It cuts the error about in half. And this blue line here is basically showing an impossibly good method where we do a scan over parameters you can't actually learn on real data. And the key point of that is that some back-of-the-envelope approximations of these regularization parameters work about as well as scanning over parameters and finding the optimum. We can't really know. So the key point here is that structural variants help you. And they don't bring this down to zero, so they don't solve everything, but they help, like many things help, and allow us to go through and study what is going on in populations of tumors. This is based on a subset of the TCGA breast cancer data. We needed to work with whole genome sequence, because if you don't have whole genome sequence, you don't have most of your structural variants, and unfortunately, most of the TCGA, we don't have that. So this is based on just 31 of the TCGA breast cancer samples where we have whole genome sequence, and essentially what we're able to show is that you can run these samples, you can get phylogenies. I wouldn't place a great deal of confidence in any one phylogeny here, but we can see a spectrum of different phylogenies across these tumors, and we can start to study statistical properties that you get out of these, so quantifying features of these tumors. And I would say this is not by any means a finished product yet, but essentially what we're able to show is that by a variety of different measures, you see interesting subclasses within these tumors. We see at least a bimodality among the structurally variant tumors in levels of heterogeneity and diversity by structural variations. And we know from our prior fish studies and other studies that these kinds of variations in the mutator phenotype are very often the really interesting things if you're trying to predict what is likely to happen next within a tumor. And I will point out that we're seeing in our data bimodality. As I mentioned, these techniques are not yet practical for the most highly structurally variant tumors, so probably there's at least a trimodality within the mechanisms of structural variation acting in these tumors, and I'd be willing to bet that's going to turn out to be an interesting thing for predicting future progression of these tumors. So one last thing I want to leave you with before I run out of time here is that another way of improving on your methods is to try to bring in different classes of data. And one of the things we've been working on for a couple of years now is ways of bringing together different subsets of these kinds of data. So fish and bulk data, and in this case, bulk and single cell sequence data. So I know there's been work published by Mlichik et al. This was a collaboration of Jenks Hinaup's group and Nico Bierenwinkel's. I haven't seen Cenk or Nico here, so I guess but I think they're representatives, at least of Nico's group. So really interesting work going on in that direction. We've been pursuing a particular version of this, again, focused on copy number variation. Again, just trying to ask the question of, can we do a little better bringing in these different kinds of data? And a lot of what has been guiding our thinking has been the fact that, at least so far, single cell sequence has been really cool, but not available in the volumes you need if you want to look at a lot of variation per tumor and a lot of tumors. And so the proposal of both their work and our own was this idea that maybe we can do a lot if we have bulk sequence and just a few single cells per tumor. Maybe you can do better than you can with just bulk sequence or with lots of single cells on a few tumors. And so what we've been working with is a kind of semi-simulated study, taking some real copy number variation single-cell sequence data from the lab of Michael Dean, and trying to ask, could we do a better job of deconvolving this, semi-simulating mixtures of these? We know the ground truth. Now, if we want to do this, we need to start with a kind of baseline method. We use just pure non-negative matrix factorization using a classic algorithm of Lee and Sung. And we're doing this on copy number variation or copy number aberration data, and we know this should not work. You shouldn't be able to deconvolve just pure copy number variation data because it's an underdetermined problem, but it's a baseline we can use as a starting point. And we can then start to throw in some heuristics to try to ask if this problem is solvable. If you just do your pure NMF, basically we see what you would expect, it doesn't work. So the accuracy of deconvolution is about 5%. It's completely useless by itself. But just throwing in a little biological knowledge, the assumption that these tumors are largely diploid, ends up bringing that up to about 50% accuracy. So just adding that as a constraint gives you a way of resolving the underdeterminedness, or to a large degree, of the problem. You can get some accuracy out of it, but we're not using the single cell data yet. But you can use a similar intuition to throw in single cell data heuristically, more or less just initializing your non-negative matrix factorization with single cells and asking, does that lead to an improvement? Just telling it, start looking around for things that look like your single cells. And the answer here is that it does. At least in the semi-simulated data, that gets you from about 50% accuracy to about 90% accuracy in your deconvolution. Now, that 10% error can make a very big difference if you're trying to learn something about the variation of the tumors, so this is not at all a solved problem. But having the single cells does lead to some significant improvement. This is just an example of one particularly good case here, where pretty much you almost get an exact deconvolution. But I want to emphasize that this is from very unrealistic assumptions about the data. This is assuming for the moment we have a lot of samples and a relatively simple tumor. And so we would then want to ask, can we improve any further on this and get to something that's going to work really well on realistic numbers of samples? Well, we showed that working with a heuristic kind of initialization with single cells helps, but really it's better to have a nice, well-defined objective function that says what you're trying to solve, and we can take this the next step by building that into our objective function. The intuition of this technique is that the deconvolved modes should look like your single cells, and we can throw in an L2 term to our objective to say, let's bias the deconvolution to do that. You can actually modify Lee and Sung's algorithm pretty simply to solve for that objective, and that actually ends up working very well, although still under unrealistic assumptions about your data. So if you assume you have lots of samples, you can get close to 100% accuracy doing that. Just saying, find me things that look in my deconvolution that look like my single cells. Now, it turns out that that is somewhat dependent on how you construct your simulated examples, and we want to take this a further step and say that even though that is a computationally convenient way of saying we want things that look like our single cells, it's not a very biologically well-motivated way of posing this. So an L2 is basically saying our deconvolution modes should, in some crude sense, a differentiable sense, look like our single cells, which is nice algorithmically, it's not very nice biologically. Really what we want to say is that things should be nice phylogenetically. The modes we're trying to discover in our deconvolution should look like they're related to the single cells we observe. And you can build that in by building a phylogeny, throwing in this case again, an L1 objective, so a very crude minimum evolution phylogeny. And it turns out that that sometimes helps you. If you're looking at samples with very low heterogeneity in them, so not a lot of divergence, it really doesn't matter how you measure your similarity. If you're looking at more divergent examples, then building a phylogeny helps you appreciably. It gives you about a 10% boost in the accuracy, although everything works worse on more divergent samples, but at least we get some improvement there. But we see that there is a significant amount of diminishing returns there. This is, I would not put a lot of stock in this result yet. This is very kind of hot off the presses incomplete results from our group, but basically what we can find is a few single cells help you in doing this deconvolution, at least the way we're measuring error here. You don't get a lot of extra boost by adding more cells. Now, I think this is to some degree an artifact that we're looking at overall accuracy, which is not necessarily what you want to look at. There are particular aspects of the deconvolution that will be important, particular variants you really care about and others you don't care about. But at least for the moment, what we're seeing is a few single cells can help you. At least if you're measuring by quality of your deconvolution, you don't get a lot of extra help by adding in more single cells. Now, all of this, though, is still under very unrealistic assumptions about the amount of data we have available. What is realistic is a moving target, and actually I'm told there are new technologies out where these are not such unrealistic assumptions, and you might actually be able to get 100 samples from multi-region sequencing now. But for the moment, what we've been assuming is that really, if you have multi-region sequencing, you're looking at maybe 1 to 10 samples if you're lucky. And so we've been looking at one other way of using the single cell data, which is to try to artificially boost the dimension of your data by essentially taking your real mixed data and throwing in some synthetic mixtures that you can build from your real mixed data and your single cell data as essentially a way of getting around the problem that you can't infer more dimensions from your data than were actually present in the raw data. And this again is a very early result. I wouldn't put a lot of stock in it, but basically the key point of this is that you can do something with this. You can use this kind of enrichment strategy to allow you to get decent results, even from purely copy number data with just a single mixed sample, although you lose a lot relative to what you would get with large numbers of regions. So essentially the key point for the moment is you really want lots of ideally, you'd have lots of regions and lots of single cells per region. That's what we want to get to, but using the data we have available right now, there's a lot you can squeeze out of it if you're a little creative about how you put different kinds of data together. So all of that is running through a number of different directions, and what I hope I left you with is the idea that this is far from a solved problem. I know others are doing better than us with the aspects of this problem. I don't want to detract from any of that, but no one has solved the problem of how to really construct the full complexity of tumor phylogenetics. But we can reach some conclusions from some of these methodological directions we've been working on. Probably the most important thing is to recognize that understanding tumors as a model of evolution is really the key to interpreting tumor genomic data. I think I can't possibly be the first person to mangle this famous quote for the cancer field, so apologies if someone else has quoted this before, and apologies to the original source of this, but I think the key takeaway is that nothing in cancer makes sense except in the light of evolution. But we are able to learn a lot using that, but for the moment there is no magic bullet. I know a lot of people are saying now single cell sequence is the answer, we'll learn everything, we'll get lots of single cells. For the moment at least, that is decidedly not the case. You learn things from fish, you don't learn from bulk and single cell. You learn things from bulk, you don't learn from single cell. You learn things from single cell, you don't learn from bulk or fish. And you have more power bringing these together with any one individually. The same applies for different kinds of variants, and the same applies for different kinds of algorithmic techniques, and the same applies for different ways of bringing all of these classes together. And a lot of the interesting work in getting us closer to the ground truth that we know about will be in the messiness of bringing all of these different directions together into common inferences. So we still need better data, better knowledge, better models, better algorithms, and hopefully some of the students here will be the people who discover these. So I will just close with some acknowledgements. I was describing threads of work from many people who have passed through my group over the years, as well as our collaborators, computational collaborators, primarily from the group of Alejandro Schaefer at the NIH, and Jan Mon, Gary Miller from Carnegie Mellon, as well as our experimental collaborators, Stanley Shackney, and the groups of Thomas Reed and Michael Dean at the NIH. And I have to thank our funders, and thanks to all of you for your attention.
When you're doing the connection part, the first part of the talk, it seemed like
Yes, it's the I'll repeat it. So the question was about our dimensionality reduction and our mixing. We're typically starting with PCA, which is assuming it has a linearity assumption built into it. So I will say that some years back we thought about that question. We tried some nonlinear dimensionality reduction methods, and nothing worked better than PCA. And I think the reason for that is that PCA is the least dependent on having large amounts of data to get decent results. So I think it was a trade-off that there are nonlinearities and we should be able to exploit those, but our data is too sparse to do a good job with the methods then. We haven't revisited that question recently, and it may be that going back to nonlinear dimensionality reduction or just building that into the manifold learning would be a better way of solving this now with some of the newer data sets. Well, the mixtures we would expect to be linear, and if you're looking at DNA data, you would expect linearity to be a pretty good assumption. When you're bringing in the RNA data, then you get nonlinearities because of communication between the cells, and probably the same is true with methylation, which we haven't worked with. And there may be other sources of nonlinearity. We're just not considering errors, noise in the technique. So it's always better not to assume linearity if you don't know it's there, but it's a decent assumption for DNA data. So really it's one big integer program. So I broke it down into sets of constraints that refer to different aspects of the problem. But basically it's one big integer program, and then you can assume that some variables are constant, try to solve it, assume different ones are constant, try to solve it. So that's more or less how the coordinate descent works. Essentially we're fixing some of the variables and trying to solve it, and fixing other ones and trying to solve it. And it's not a guaranteed optimum method. That's the kind of strategy we're pursuing.
Okay, so let's